are special human cells that have the ability to develop into many different types of cells, from muscle cells to brain cells. In some cases, they have the ability to repair damaged tissue, like this. Stem cells originate from two main sources: adult body tissues and embryos. Scientists are currently working on ways to develop stem cells from other cells using genetic reprogramming techniques. Researchers believe that stem cell-based therapies may one day be used to treat devastating ailments like paralysis and Alzheimer's disease. Stem cells are divided from two main forms: embryonic stem cells and adult stem cells. The embryonic stem cells used in research today come from unused embryos, resulting from an vitro-fertilization procedure that are donated to science. The three main types of stem cells are totipotent, pluripotent, and multipotent stem cells. Totipotent cells can form from all cell types in a body, plus extra-embryonic or placental cells. Embryonic cells, when the first couple of cell divisions after fertilization, are the only cells that are totipotent. Pluripotent cells can give rise to all cell types that make up the body. Embryonic stem cells are considered pluripotent. Multipotent cells can develop into more than one cell type, but are more limited than pluripotent cells. Adult cells and cord blood stem cells are considered multipotent. As previously mentioned, stem cell research can yield numerous medical benefits in areas of regenerative medicine, including potential treatments for Alzheimer's disease. What is Alzheimer's disease? Alzheimer's disease is the leading cause of dementia. People affected by Alzheimer's disease commonly experience memory loss, confusions, and severe mood swings. We still don't know why or how Alzheimer's disease comes about, but several studies focus on two proteins in particular: amyloid beta proteins and tau proteins. They are both found in deteriorating areas of the brain affected by Alzheimer's. The reason that amyloid beta stem cells can be disastrous is because if they clump together too much, they can become toxic. Abnormal levels of amyloid beta cells in Alzheimer's brains clump together to form plaques that collect between neurons and disrupt cell function. Currently, research done by organizations like the Alzheimer's Foundation of America and Banner Alzheimer's Initiative are researching what happens when these clumpings occur. Tau proteins are other proteins that usually are found attached to microtubules in order to guide cell function in the neurons. In Alzheimer's disease, abnormal chemical changes cause tau to detach from microtubules and stick together with other tau molecules. This eventually forms a tangle inside of neurons that block the neuron transport system, disallowing communication between neurons. And even though we have all this knowledge on the Alzheimer's disease and proteins within it, there is still no known cure for Alzheimer's disease. However, stem cell research is emerging as a new way to combat Alzheimer's disease. Although there is currently no form of stem cell research of Alzheimer's being tested on humans, positive effects have been seen with neural stem cell transplants given to mice with very similar diseases to Alzheimer's. Right now, researchers are studying what these stem cells are doing and how they might repair the brain in humans. Neural stem cell transplants have occurred before. Nayan Gupta of UCSF Med used this technique when attempting to treat Pelizaeus-Merzbacher, a disease, which is a genetic malady that leaves neurons without their myelin coating. When doing his neural stem cell research, he used very similar techniques that were used on the mice during the Alzheimer's research. Within his operations, he took pluripotent stem cells that have the ability to self-renew, into the three primary germ layers of the early embryo, and he took those pluripotent stem cells from the brain of aborted fetuses. In the lab, he expanded these cells and injected them into the brains or the spinal cords of patients with these incurable neurological diseases. The same would be done on humans with Alzheimer's. There are many challenges that come about when trying to use stem cell research to combat Alzheimer's. First and foremost, every single case is unique and very difficult to treat. Large parts of the brain have been destroyed during Alzheimer's. Due to the fact that there has been little testing done, it is difficult to tell if it will work out. But this is a major step in the right direction for combating Alzheimer's disease with the help of stem cell research. Debates over the ethics of embryonic stem cell research have divided scientists, politicians, and religious groups for years. Many parties continue to have strong opinions that trigger ongoing debates about stem cell research, and the following pros and cons could provide a snapshot of some of the points on each side of the issue. The excitement about stem cell research is primarily due to the medical benefits in areas of regenerative medicine and therapeutic cloning. Stem cells provide a huge potential for finding treatments and cures to a vast array of medical issues. On top of that, there's an endless potential for scientists to learn about human growth and cell development from studying stem cells. For example, by studying how stem cells develop into specific types of cells, scientists potentially could learn how to treat or prevent relevant ailments. One of the areas of potential is embryonic treatment. This stage of pregnancy is when many birth defects or other potential issues begin. Studying embryonic stem cells could possibly lead to a better understanding of how embryos develop and maybe even lead to treatments that can identify and address potential problems. But, like any other form of research, stem cell provides problems. And most opposition to stem cell research is philosophical and theological, focusing on questions of whether we should be taking science this far in the first place. It's not easy to obtain stem cells. Once harvested from an embryo, stem cells require several months of growth before they can be used. Obtaining adult stem cells, such as from the bone marrow, can be very painful. On top of this, the cost can be prohibitive for many patients, with a single treatment costing well into thousands of dollars. The use of embryonic stem cells for research involves the destruction of blastocysts, formed when in a laboratory fertilized human eggs. For those who believe that life begins at conception, the blastocyst is a human life, and to destroy it is unacceptable and immoral. A similar theological problem is an idea of creating living tissue in a laboratory and whether that represents humans taking the role of God. This argument also applies to the potential for human cloning. For those who believe God created people, the prospect of people creating people can be very troublesome. My opinion on the subject is that we should be continuing to develop stem cells for as long as possible, because even if there is the slightest chance that we can cure diseases like Alzheimer's, we should take it. And yes, there is the argument that we are playing the role of God. However, the more research we have done and the more knowledge we obtain regarding stem cells, the better able we will be to create synthetic stem cells that take out the argument that humans are playing the role of God when creating stem cells. Thank you for listening.